We're learning about the mystery of the universe here, right? Learning about the mystery of the universe. I'm hoping one of you will. This will be the foundation that you use to change the world. Hopefully, I'm not smart enough to do it. Somebody in here is, though, I'm sure. Okie dokie. Well, let's move right along. Okay, cytology. So in lab, we talked about eukaryotic versus prokaryotic cells. I don't really know that I need to redux that. We talked about what makes animal cells in particular so different. Their cell membrane's flexible, right? It's flexible, has this cholesterol in it. That is very different from plant, right? Where plant cells tend to be very rigid. Some animal cells can get around with flagella, right? You guys know what a flagellum is. And what are cilia? Do our cells use cilia to get around? No, they use cilia to move substances on top of them, right? Like mucus blankets and things like that. Okay, we talked about the cell membrane. Let's see. You guys are okay with the cytoskeleton, the centrosomes, and the centromeres, cytoplasm versus cytosol. You guys okay with that? The difference between cytoplasm and cytosol?
Do you mind recapping that really quick?
Yeah, so cytoplasm is everything, right? It's all the organelles in the fluid. Cytosol is just the fluid part of it.
Inside the cell.
Inside the cell, yeah, intracellular. We talked about the nucleus. The nucleus contains nucleoplasm, okay, where you have your chromosomes. And then the nucleolus, of course, is not bound by a membrane, okay? It's just kind of in the center of the nucleus, and it contains the what? For the ribosomal RNA, right? We also have endoplasmic reticulum, right? You have two flavors. You have rough and smooth. The rough contains what? Ribosomes. We know what ribosomes do. The smooth does not contain ribosomes. The smooth ER is kind of like a factory or a warehouse, right? And this is where lipids are produced and metabolized. This is where you contain enzymes for detoxification. I talked about drugs, right? I talked about medications. And I talked about most of our medications that we take are metabolized via enzymes that exist within the smooth ER, particularly in liver cells that we call hepatocytes. Let's see here. Ions are stored in there as well, sodium, for example. Golgi apparatus, we talked about that as well, right? It contains many different Golgi bodies, and this is kind of like the post office of the cell. So as those proteins are created, they're kind of packaged in the Golgi apparatus, and then they're sent out to wherever they need to go in the cell. You guys okay with that? You also have some ribosomes floating around in your cytoplasm as well, so they're free ribosomes. And then you have these little sacs, these little membrane-bound sacs that contain enzymes. You have lysosomes, right? These help break down wastes. Certain cells use them to destroy bacteria, destroy certain pathogens, bacteria, parasites. Related to them, just in general, they're called vesicles, and then you have specialized vesicles like lysosomes and etc. So I don't think I need to really talk about that. So let's just go ahead and concern ourselves with the mitochondria, because we did not talk about the mitochondria in lab, did we? Or not specifically. Okay. So the mitochondria is kind of it's a kidney bean shaped organelle, and it has a double membrane. And the major role of the mitochondria is what? Producer of ATP. And what is ATP?
Energy.
Yeah, it is the cellular energy. I say energy, okay, energy is roughly described as the ability to do work or to do stuff. Okay, that's what energy is. And we have two major forms of energy, right? Two major flavors. We have potential energy. All right, potential energy and kinetic energy. And what is kinetic energy? It's the energy of what?
Motion.
It's energy of motion. So something in motion has kinetic energy, right? Anybody who's taken physics, you remember that that formula, right? The famous formula, kinetic energy equals 1/2 mv squared, right? The mass times the velocity squared divided by 2, half of that. So it's the energy that something in motion has, okay? Well, what's potential energy? What is potential energy? That's something a little different. What is that?
The potential to move.
Stored. Yeah, think of potential energy is kind of like the bank. Potential energy is stored. All right? It is energy, it's stored energy, and when it's used, it can become kinetic energy. So for example, if I take this marker here, and I put this marker up here, okay? Put it up on a hill, for example. Now, is it moving right now? No. Well, at least not relative to us. It's not moving. But does it have the potential to move?
Yes.
Yes. Right? It has energy stored in it. And all I need to do is put a little bit of energy into it, and what will happen? It'll fall, right? And the gravitational potential energy will be converted to kinetic energy as it falls down. Does that make sense? And that's kind of like a chemical reaction, right? Here up on the hill I have, okay, some molecule, and I want the molecule, some reaction to happen. And so what do I need to do?
Enzyme.
Right? Or I need to activation energy, enough energy to push it over the hill so it falls. So activation energy falls, and the potential energy that was stored up in there is turned to kinetic energy. You guys okay with that? So it's very similar in biology, where what we do is we take energy and we store energy in what we call high energy molecules. You guys okay with that? High energy molecules. And the high energy molecule in the body is adenosine triphosphate. And all it is it's an adenosine molecule that has 3 phosphate groups, okay, attached to it. And the way that I think of adenosine triphosphate is I think of it as, let me draw here. It's not going to be a great drawing, but I think of ATP as kind of like a gun, okay? Kind of like a gun, right? There's a bullet in the gun. There's potential energy stored in that gunpowder, right? And when that gunpowder ignites, that potential energy becomes kinetic energy, pressure builds up, fires a projectile of the gun. It's somewhat similar to ATP. And you can think of an ATP as kind of coming in and you pull the trigger on the ATP, and this 3rd phosphate group flies off. Okay, and as it flies off, you have energy that is released. And that energy can be used to power protein pumps and power chemical reactions and power cellular processes and cause muscles to contract and relax, and etc. Okay? So it is the ATP becoming ADP, adenosine diphosphate, right? And that release of energy when it becomes diphosphate is what is going on there. Does that kind of make sense? Yeah? So it is the potential energy stored in that ATP that is converted to kinetic energy, okay? The potential energy is not gravitational, it's in the chemical bonds themselves, right? You guys okay with that? Does that kind of make sense, more or less? Okay, cool. So, this mitochondria is kind of important when it comes to producing ATP. And how does our body produce ATP under normal conditions? Well, Under normal conditions, this is what happens. All right. So this is extracellular, this is intracellular, here's my cell membrane. In normal conditions, what molecule is involved? Before we get to ATP, what molecule is involved? Say it? Glucose. Glucose. And this is the amazing thing, guys. This is so amazing when you really think about it. If you pull back and you look at the big picture, the big picture is I've got the sun. And I've got a little blue planet orbiting that sun. A little blue planet orbiting that sun. And that sun is releasing lots of energy, right? All the sunlight coming off of that sun, that has energy, right? And that energy hits the planet. And then when you look on the planet, you've got a plant. Right? You've got a plant. And that plant takes the energy of that sun and uses that energy to make sugars, right? Starches. Starch is just a complex sugar, right? It's a complex carbohydrate. And then animals eat the sugars. So the energy from the sun goes into the plant, right? And it's stored in potential energy. And then we animals eat that plant, okay? Or maybe a cow or some animal eats that, and then another animal eats that animal. But can you see how it's all coming from the sun? All that energy at the end of the day is it's going from the sun into the planet, into the plant, into your mouth, into your body, and it's turned into these high energy molecules. That's a big picture. And that's just so amazing when I think about it. It's wow. It's just energy is just being transferred from the sun in several steps to us. Okay, so we've got this glucose here. And glucose, okay, so you've eaten the plant, you have metabolized those sugars into simple sugars like glucose. And that glucose is floating around in your blood, and it gets to a cell that needs to make some energy. So the glucose is going to enter the cell through the membrane, okay? And there's actually a molecule that will help the glucose enter the cell, okay? And that molecule is called insulin, okay? Insulin will facilitate the diffusion of the glucose into the cell. And right here at the membrane is the first of 3 chemical processes that occur, okay? And this, does anyone know what this first process is called? Glycolysis. Glycolysis. Yeah, good. Glycolysis. And what happens in glycolysis is we kind of break that glucose down a little bit. We break that glucose down into pyruvic acid. And you may hear the term pyruvate. You may hear the term pyruvate. And anytime you see 8, basically what that means is that an acid has done what an acid will do, and that is what? Give up a hydrogen ion. So when an acid gives up a hydrogen ion, we'll sometimes call it an 8. So lactic acid, for example, if lactic acid gives up its hydrogen ion, we call it lactate. There you go. Yeah. In chemistry, because you all have to take chemistry, in chemistry we would say that this is the conjugate base of the acid. But you guys will get there in your chemistry classes. Don't worry. So, do I make any ATP in glycolysis? Is any ATP created? No? Actually, yes. 4 ATP molecules are produced, but 2 of them are used to run glycolysis. So you're only netting at the end of the day 2 molecules of ATP per pass at glycolysis. That's not a whole lot, is it? That's not a lot at all. Now, the cool thing about glycolysis is no oxygen's needed. Okay, this cycle is what we call anaerobic. Anaerobic means without oxygen. Now, there are some cells that can get all their energy through what we call anaerobic respiration, just through glycolysis. Okay, there are some cells that can do that. These are very specialized cells. One of them happens to be the red blood cell. Red blood cells are very unique. They don't even have a nucleus. They don't contain DNA. So red blood cells can't even make proteins. So red blood cells are basically a cell membrane surrounding a bunch of hemoglobin, right? And so basically all they need to do is float around, transport oxygen. So they don't need a whole lot of ATP. So red blood cells are okay, but any cell that does anything else, if a cell makes protein, right, if it's involved in some job where it's using energy, okay, anaerobic respiration is not going to be enough. So this is where the mitochondria comes into play. That pyruvic acid is going to go into the mitochondria. It's going to go through two different cycles. The first cycle that it's going to go through is something called the Krebs. The Krebs cycle. Okay. Another term for that is the citric acid cycle. Okay. Or the tricarboxylic acid cycle, the TCA cycle. Okay. All three names mean the same thing, but the common term is the Krebs cycle. Now, in the Krebs cycle, what we have here is we have basically going into the Krebs cycle, you have a little carbon backbone, and then attached to those carbons, you have hydrogens. And what you start doing is you start taking those hydrogens off. You start tearing those hydrogens off of the carbon. Okay? And those hydrogens, as they get tore off, okay, there are transport molecules, FAD and NAD, and they transport those hydrogens to another cycle that we'll talk about here in a minute. Okay? And so what I have left over, once all those hydrogens are tore off, basically I have carbons and oxygens left over. You guys cool with that? And those carbons and those oxygens will combine to form CO2, or carbon dioxide. You guys okay with that? This is where the carbon dioxide waste is produced. Right? You exhale CO2 as a waste. This is where it's produced. It comes out of the Krebs cycle because we've torn these hydrogens off of this carbon-oxygen backbone here. All right? And that's where our CO2 comes from. So you've got CO2 coming out of the Krebs cycle, and you've got this NAD and FAD with these hydrogens attached to them. And they dump those hydrogens off into the last cycle. And this last cycle is known as the electron transport chain. Right? Electron transport. The electron transport chain. Some people call this oxidative phosphorylation as well. Okay. And what happens in the electron transport chain is within the inner folds of the mitochondria, okay, within the membrane. So here I have my membrane, and I'm just going to draw some proteins going through the membrane, like so. All right. So what happens is the NAD and the FADHs drop those hydrogens off on this end of the membrane here. And these hydrogens are in their ion form, so they're all H pluses. All right? They're all H pluses. there. It's such an important thing. I don't want all that noise coming in. Okay, these are all H pluses, and all those electrons, all the electrons that have been taken from those hydrogens, those electrons, the energy that those electrons have, because they're at higher energy, the energy that those electrons have is actually used to power these proteins here. They're called cytochromes. All right? And so these electrons start at high energy and then they move to low energy as they go through and help power those proteins. All right. And then right here at the very end, there is a protein called ATP synthase. Oh, that's an enzyme, isn't it? And what do you suppose ATP synthase does? It synthesizes ATP. How about that, right? So here is the crazy thing that happens. This is like a dam. All these hydrogen ions are piling up here. And this protein here has a little channel that goes through it that allows those hydrogen ions to go through it. All right? And then it kind of has these little projections that come out of it. And it has an ADP here plus a phosphate, okay? And what happens is as these hydrogen ions flow through that channel, they literally cause the channel to turn like this as they flow through, like a turbine, like a dam, right? Like a hydroelectric dam. And as that spins around, okay, it actually attaches the energy, okay, the energy of it spinning around attaches that third phosphate to ADP and creates ATP. Does that make sense? That's something called proton motive force. So it's literally like a dam inside of your mitochondria, pumping out all this ATP.
It's a literal powerhouse.
It is. It is literally, yeah, it is literally like a hydroelectric dam inside of your mitochondria. All right? But here's the thing. Now you have all these hydrogen ions, right? All of these hydrogen ions that are collecting down here, right? All these hydrogen ions and then all these electrons at lower energy levels. I need to get rid of this somehow, right? Because if these pile up, then I lose my gradient, right? And then the flow stops. So I need some way of getting rid of all this stuff. How do you suppose I do that? No? This is where oxygen comes in. This is why we need oxygen. Oxygen comes in, and oxygen can combine with these hydrogen ions and electrons to produce H2O, to produce water molecules. So the way I look at oxygen is an intracellular garbage truck. It comes in and it gets rid of all this garbage that piles up. That's why you need it. And so this here, electron transport, is what we call aerobic respiration. Okay? And you produce 28 to 34 ATP molecules per pass here. So significantly more here than in anaerobic. You guys okay with that? So this is the super simplified version, okay? You guys, in other classes, you may go into more detail because each of these cycles has multiple steps. I'm not going to make you memorize the steps, but you do want to know the 3 cycles and in general what occurs. Okay, because this is how we make energy, and we haven't known this for very long. Okay? We've known this for about as long as I've been on this earth. Okay, so more or less 4 decades. Okay, we didn't really figure this out. This is called the chemo-osmotic theory. We really didn't get this nailed down till the 1970s. Okay, so That may seem like a long time for some of you guys, but it's really not. Really not a long time at all. Okay, so you guys okay with that? You okay with the mitochondria? What's going on there? Just in general, okay, this is big picture, what's going on. Okay, now what I want to do, you guys got this? Unfortunately, I have to erase it, I'm sorry. But you guys you okay there? So I'm going to erase this, and now what I'm going to do is I'm going to talk about probably the concepts that gave the people the hardest time in lab, okay? The hardest time in lab. And these were things that we didn't really specifically talk about. Okay, and this is what's going on with all that water, all that solvent and solute stuff. All right, so here I have my cell membrane, right? And is the membrane permeable to everything? No, the membrane is what we call what? It's not semi, it is selectively permeable. It's selective. So it's selectively permeable. It's able to select what can go through it and what can't. In general, large molecules can't go through it, right? Large dissolved molecules. So this is intracellular and this is extracellular. Okay, outside and inside. Now, what I can do, can water go through though? Can water freely pass through the cell membrane? Yes. Yes, it can pass through those aquaporins, right? Now, what I can do is, first of all, are you guys okay with, are you guys all on the same page of that not all solute can pass through the membrane? So let's say, that I have this cell here, and outside of the cell I have a lot of solute dissolved. So the solute concentration is very high. Alright. And there's just a little bit of solute inside of the cell. Now, where would this solute like to be? It'd like to get into the cell, right? And solute moving from high concentration to low concentration is what? Diffusion, right? Diffusion, right? Diffusion is the movement, going back to lab, diffusion is the movement of solute, okay, from high to low solute concentration. Now, let's say that this solute cannot get through the cell membrane, okay? What can move, though? Water can move. And a special type of diffusion is known as osmosis, right? Osmosis, you guys okay with that? And osmosis is not the movement of solute, but rather the movement of solvent, aka water for biological purposes, from where? A solvent moves from
High concentration.
low solute concentration to high solute. So water is going to move toward the solute, okay? So if I have a situation where I have lots of solute outside of the cell and a little bit of solute inside of the cell, where is the water going to move? It is going to move from the inside to the outside. The solute produces a kind of pressure, what we call osmotic. Osmotic pressure. Solute causes osmosis because it can't get through the membrane, but water can. So when I have a high concentration of solute outside of the cell, it's going to pull water outside of the cell with it. Are you guys okay with that? Cool? All right, so what I'm going to do is I'm going to get rid of these. And what I'm going to say is I have a situation where I have roughly the same amount of solute, concentration of solute outside of the cell as inside of the cell. So the osmotic pressure inside of the cell and outside of the cell is going to be the same, right? You guys okay with that? So what way, if you had to guess, what way is the water going to move?
Both.
Yeah, is some water going to move out of the cell?
Yes.
Is some water going to move into the cell?
Yes.
But it's going to move at an equilibrium. So the net movement of water in and out of the cell is equal, right? In this situation, where outside of the cell is the same as inside, the solute concentration of the osmotic pressure, this is what we call an isotonic. An isotonic situation, okay? An isotonic or a balanced situation. And it has to do with solute, right? The concentration of solute is roughly the same outside as inside. You guys cool with that? That's isotonic. So if I were to expose cells to an isotonic solution, would the cells get bigger or smaller?
Same.
They'd stay roughly the same, right? Now, let's go back to my original situation. Let's say that I have somebody who's very sick and they have a very high concentration. Maybe they have something called hypernatremia. Hypernatremia is high sodium in the blood. So they have a high amount of sodium outside of the cell compared to inside of the cell. All right? Now, what way is the water going to move?
Outside.
The cells are going to become dehydrated. Water is going to move outside of the cell, so the cell's going to get bigger or smaller?
Smaller.
They're going to shrink or they're going to undergo crenation. Not cremate, but crenate, crenation. They'll shrink. And this is a situation that is hypertonic. The cell, so tonicity, to say tonicity, you always have to have a membrane involved. Okay, it's the difference between one environment and another. A hypertonic environment is where the environment outside of the cell has more osmotic pressure, is more solute concentration than inside. So the water is going to go where? Out. It's going to get pulled out of the cell. So if you were to inject a hypertonic solution into your patient, if you had a patient, you would start pulling water out of their cells. Does that make sense? Okay. Now, let's say that you guys are really frustrating me, and I'm not feeling the love. I'm not feeling the love at all. And so I want to go out and have a good time. How am I going to go out and have a good time and feel really, really good and feel a lot of love? I'm not going to drink. No, come on. You guys are young, hip, right? How do you do that? Huh? No, I'm going to drop some X. That's what I'm going to do.
Wow.
I'm going to go out and drop some X. Right? Some Molly, some MDMA, right? Some ecstasy. Right? That's what I'm going to do.
Wow. It's been a good time.
Yeah.
Crazy.
Right? I'm having a great time. I'm feeling the love now, right? But that ecstasy, that MDMA, well, it has some effects on me. And one of the things that it can do is it can alter the way your kidneys work. And so what happens, in my case, okay, is I end up losing a lot of solute. And so now I have a situation where the solute concentration inside of my cell is higher than the solute concentration outside of the cell. Where's the water going to go? The water is going to go into the cell. So what's going to happen to my cells?
Swell.
They're going to swell, right? They're going to swell, and if they swell enough, they may even rupture or lyse. They may even rupture or undergo lysis. Okay? And this is what we call hypotonic, where the concentration of solute outside of the cell is less than inside of the cell. So hypertonic, the cells become dehydrated, they crenate, they lose water. Hypotonic, the cells swell, okay, they take on water, okay. And one area where this is really bad is in your brain, because your brain doesn't really have anywhere to swell into, right? It's surrounded by a hard skull. So, and we have actually seen several people die that were on ecstasy, okay, because ecstasy makes you, MDMA, Molly, whatever you want to call it, it makes you very thirsty, one, okay, it makes you thirsty, and so you drink a lot, and you end up not urinating. It makes you thirsty, and then it interferes with some hormones that help you pee, specifically something called antidiuretic hormone, or vasopressin. So you're drinking a lot of water, you're not peeing, and you end up diluting your blood out. Your blood becomes hypotonic, and then your brain cells begin to swell, and then you start having seizures, and you potentially die. We've seen several cases of this occur. Some people have even called this quote-unquote water toxicity. You guys ever heard of that term, water toxicity or water intoxication? And this is one way by which you can make drinking too much water really toxic is by taking something like MDMA that interferes with your kidneys. Yeah?
So then why is it that when you go to the hospital, let's say someone's to go to the hospital for like, they were on X, and they give you fluids, why would they be doing that knowing that it could possibly kill you?
Well, what type of fluid are they giving you? They're probably more likely giving you an isotonic fluid, not a hypotonic fluid. So they're going to give you an IV fluid that is more balanced. They may even, if it's really bad, if your sodium is really low, they may even give you a concentrated sodium solution. They may even give you something called hypertonic saline to help increase the sodium level in your blood to treat that. Yeah. Now, not everybody that does ecstasy is going to get this, of course, but this is just one example of where this can be a real big problem. This we call hyponatremia, low sodium levels in the blood, as opposed to hypernatremia. Okay. Yes, sir.
Sorry, real quick.
Yeah.
So just in this case, the cells are retaining more just because of the solute that's in there. So would it technically, in isotonic solution, now be a hypertonic solution?
Yeah, so an isotonic solution, well, it's going to be balanced with the cells, though, more or less. But it's going to be more well, not really, because remember, tonicity has to do with inside and outside of the cell. So if I'm injecting a solution that is hypotonic or isotonic to the cells, it's still isotonic. The solution itself is, at least, that you're giving. Yeah. Absolutely. So are you guys okay with the difference between hypertonic, isotonic, hypotonic? Does that kind of make sense? Now, your cells have some very special proteins that allow certain ions to get in and out of them. Okay. One of the most important proteins is something known as the Sodium-Potassium Pump. And I believe the book talks about this a little bit. The Sodium-Potassium Pump. And what the Sodium-Potassium Pump does is it pumps sodium out of the cell and pumps potassium into the cell. Okay?
Sodium out, potassium in?
Sodium out, potassium in. Now, normally, diffusion is what kind of process? Is diffusion active or passive? It's passive, right? It's going with a gradient. So diffusion is if I have a lot of sodium here and just a little bit of sodium here, okay, the solute's going to diffuse out. That's passive, right? But in the case of this sodium-potassium pump, okay, so I've got my cell here. I already have a fair amount of potassium inside of the cell. Okay, and I have a fair amount of sodium outside of the cell. Maybe I'll do the sodium in a different color, just so you can see the difference here.
Is it a potassium ion?
Yes. Yeah, these are all ions, yeah. So I have a situation that looks kind of like this. Now, where does the sodium outside of the cell want to go? In, right? Because the solute is more concentrated out than in, right? Where does the potassium inside of the cell want to go? It wants to go out. But what the sodium-potassium pump does is it pumps the sodium in and the potassium out, and it goes against the concentration gradient, okay? So is that a passive process now? No. In fact, you have to use energy, right? This uses energy. Diffusion doesn't use energy, but this does, right? This is what's known as active transport. Okay? This is a type of active process. So you're using energy. Okay? And what we can do is you can create a gradient where you have lots of one ion inside of the cell and a lot of another type of ion outside of the cell. Okay? You can create what's called a membrane potential. And all a potential is it refers to potential energy. Where you have a high concentration on one side, low concentration on the other side, where does it want to go? It wants to move to that low concentration, right? And so what we can do is we can create a situation in a cell where there's lots of sodium outside, so this is out, lots of potassium inside, and if I had a channel, another protein, not a sodium-potassium pump, but another channel that opened up and allowed sodium to flow, where do you think all that sodium is going to go? Out or in? So this is out and this is in. It's going to rush into that cell, right? It's going to rush into the cell.
Through that pump.
Through that, not the pump, through a channel. Okay. So this is what we call a sodium channel. Okay. But the sodium-potassium pump sets us up for this. So sodium-potassium pump pumps the sodium out, pumps the potassium in, and then if I open a sodium channel up, all that sodium will rush back into the cell. Certain cells use this to their advantage. Certain cells that can conduct impulses. Nerve cells, muscle cells use this. And we'll go into this in a lot more detail, but this is the foundational knowledge behind this. Okay? They can create what's called a membrane potential, and then that potential can change when other channels open up. Okay?
You said nerve cells?
Nerve and muscle cells, yeah. And this is the basis behind how muscles contract. Okay, it's the beginning of a muscle contraction, and is the beginning of how neurons can send and receive signals. Okay, this is the foundation behind all that stuff occurring. Okay, you guys okay with that? Okie dokie. So we talked about that. We talked about active transport. I think we're doing good. Oh yeah, let's talk about Pinocytosis, Phagocytosis, Endocytosis. These are like active transport, okay, in that they are mechanisms by which cells can bring stuff into them, okay? So let's just talk about these real quick. So Pinocytosis is kind of an easy one to think of. Pinocytosis literally means to drink. Literally means taking a sip. And so what happens in pinocytosis, and actually the book does a pretty good job of kind of explaining what's going on, is the cell membrane kind of forms these little indentations like this, and then solute can kind of come in, and then it'll close the indentation off, and then that stuff's in the cell. So it's kind of comes up and does a little sip. Now, that is different from what's called phagocytosis, because pinocytosis is kind of like, I'm going to take a little sip, take a little sip. Phagocytosis is literally the cell coming in, opening up, engulfing something, engulfing a molecule. Oftentimes we see this with white blood cells engulfing bacteria. Literally engulfs the whole thing, sucks it in, and then generally dumps a bunch of enzymes on that to kill it. And we talked about that in a little more detail in lab. So that's phagocytosis, and I think of that as eating. So pinocytosis is kind of like, you take a little drink. Phagocytosis is actually eating and munching down on it. And then exocytosis is just what?
Spitting it out.
Yeah, getting rid of it. So eat, drink, and the equivalent of pooping, right? All that waste is surrounded in a vesicle. That vesicle makes its way to the cell membrane, merges to the cell membrane, and then it opens up and all that stuff gets dumped out. So there you go. All right?
And that was what again?
Exocytosis. Yeah, exiting the cell. So you guys okay with that? So far, so good? All right, so we've almost gotten through this. How are we doing on time? Yeah, we're doing okay on time. So let's just, the last thing I want to do is I want to review the cell cycle, and we did this in lab, okay? But let's just review it one more time, because it can be a little confusing. And I actually already talked, I think the very last thing the book talks about is cancer. And I already kind of primed you guys on that. I probably won't get to cancer today. So we'll pick that up next week. But we're actually in a really good spot. So, all right. So let's talk about the cell cycle. So here I have my daughter cells that have been made. Okay? My daughter cells, they are identical, right? So once the new cell is made, the new cell can do one of two things. What can it do?
Divide or not.
Right. Can get ready to divide again or not. If it doesn't divide again, it enters what we call the G0 or G0. Yeah. So if it's like a neuron, like a nerve cell, okay, more likely it's going to be in the G0 phase. You guys okay with that? However, if it does get ready to divide, it will enter the G1 phase, right? And what happens in G1? That's growth 1, right? And what's the big thing that happens there? Say it again?
Duplication
Of what, though?
of the organelles.
There you go. Remember this. It is the organelles. The organelles themselves are duplicated. So you have organelles and protein synthesis. That's what occurs in G1, right? And then what happens after G1? We enter the S phase. Okay, what is the S phase? Primarily. The organelles, S is what? Synthesis. There you go. And the big thing that happens in S is, say it again? The DNA. So the chromosomes are copied, right? You make copies. And how many chromosomes do you have?
20.
And so you have how many at the end of S? 40. There you go. Yeah, they're copied, right? You make copies. Okay, so DNA copied. All right. And then the next phase is G2. What happens in G2?
Mitosis.
Not mitosis. It stands for growth 2. What's the big thing that happens there? It has to do with here. Because this is almost perfect. Not quite.
With the whatever little mistakes are.
There you go. Error correction. I just think of it as error. There are actually there are a few spots where this happens, but that's a major thing. All right, and then at the end of G2, we actually enter the cell division process itself, right? And so I'll draw that as like this. What are the steps of cell division? What's the first one?
Prophase.
Prophase. And what's the big feature that we see happen in prophase?
The nuclear envelope starts to...
It dissolves. Cool. So the nuclear envelope dissolves. Starts to go away. Okay, and then as it dissolves, we move into what phase?
Metaphase.
Metaphase. And what's the classic thing and characteristic thing we see in metaphase?
They start to line up in
The
metaphase.
chromosomes, right? So we have the centrioles, right? The centrioles, the centrosome divides one centriole on one side, one centriole on the other side, right? And then you have what? These little extensions that come out, attach to receptors on the chromosomes, and line them up down the midline. And so I think of metaphase as being the midline. It's very characteristic, very easy to see if you see it on a microscope slide. Okay. All right, and then what? Anaphase. And what happens in anaphase?
Starts to pull to the opposite.
The chromosomes are pulled toward different sides, right? Pulled away from the midline. And then finally, what do we have?
Telophase.
What happens in telophase?
Cleavage.
You have that cleavage, furrow, and the nuclear envelope begins to reform, right? And then it is not, so what is this collectively? What are these collectively known as?
Or mitosis.
This is collectively known as mitosis. And then finally, we have the cells splitting, and that is known as? All right. And from the end of cytokinesis all the way to the beginning of mitosis is known as? Interphase. So interphase includes G0, G1, S, G2. All right. That's it. That's the cell cycle right there. I think we're in a good spot for next week. I will see you guys on Thursday for lab. In lab, this is going to be a critical one for you guys. This is histology. This is going to be one of the more difficult labs, simply because there's so much stuff you're going to have to memorize. So we're going to be looking at a lot of tissue slides. And I'm going to be doing my darndest to give you all the hints and dirty tricks to memorizing what types of tissue look like what. But we'll do that here in a couple days.